Good morning, students. My name is V. Jyoti, lecturer in zoology. I am from Government Degree College from Women for Women. Okay, today topic is genetic linkage and gene mapping. Okay, first we go to introduction. After detailed study of meiosis, we came to know that not all traits are independently asserted. There are few traits which are responsible to stay gather generation to generation. These traits governed by traits is known as genetic linkage or gene linkage or gene set for genes present in whole chromosome is known as linkage group, and mapping of these genes is known as gene mapping. Okay, next introduction will say what history. Linkage is defined as the tendency of two or more genes to remain together in the original combination in the same chromosome. Linkage as discovered by T.H. Morgan. Okay, linkage has found in Drosophila, sweet pea, maize, man, etc. An organism contains many characters in its body. Each character is determined by a pair of alleles or gene. The gene for different characters may be either situated in the same chromosome or different by a pair of alleles. Okay, either that some chromosome are different chromosome when the gene are situated in different chromosomes. Okay, history. In 1906, Bateson and Pennett first evidently discovered effect of linkage in dihybrid cross in pea plant using traits like flower color and pollen shape, but they did not interpret their result in terms of gene behavior. In 1910, T.H. Morgan had successful in discovered of phenomenon of linkage using Drosophila traits like mutants yellow body with white eye and wild type gray body with red eye. Okay, it used to T.H. Morgan in first Drosophila and maize, man, sweet pea, etc. Some experiment is successful. Okay, if two genes are on different chromosomes, if two genes are on different chromosome, Mendel theme. This Mendel theme to observe this. Okay, this type of Mendel theme. Okay, next test cross to observe half look like them got a set of the parent, half look like they got a set of a parent chromosome, and half look like they got a mix of both parents chromosomes. Okay, to observe this diagram. Okay, this P for parent to release gametes. After gametes it form meiotic developed is called F1 F1 stage is called family 1 and one is testers. Some parental type 1 by 4 we observe, and after it test cross progeny 1 by 4 parental type 1 by 4 recombinant 1 by 4 recombinant we observe this total is 1:2 observe this test cross to take TH Morgan. Okay, next now suppose both gene A and B were next to each other on the same chromosome linkage gene. Okay, what happened to the ratio in this diagram? Okay, we observe this diagram. Okay, this diagram which ratio will become. Okay, this okay, next is meiotic chromosomes and meiotic products. Some product we observe meiosis with no crossing over between the genes in first diagram. Then we observe second diagram meiosis with a crossing over between the gene. So it X type to form to is crossing over is yellow and some light brown to crossing over gene. This we observe this diagram. So meiotic product will be crossing over in second part of next we observe first is no crossing over between the gene. So parental will be normal all position, but in crossing over in between the genes, so parental and recombinant will be formed and new characters are observed. Okay, okay, these some parental P4 parental gene, it gamete formation is haploid. Parental gene commonly diploid, it come haploid is called gametes. We observe capital A, capital B, and we observe brown color, small a, small b is mother and father will be separated in color. So this two different color. This cross hybrid family 1 to form zygote. Okay, we observe this first one is capital A and capital B, another one is small a, small b is cis hybrid is called recombinant, also say cis hybrid is F1 and this is tested similarly formed to yellow. After it is small a, small b, small a, small b, we observe this diagram. Then we go to test cross progeny. Test cross means anyone will be 1 by 4. 1 by 4 will be formed. This is some observation is test crossing. Parental type is capital A, capital B and small a, small b. After a parental type also small a, small b, small a, small b. But we observe recombinated capital A, small a and small b we observe. Another is less than 1 by 4, less than 1 by 4 is parental type, greater than 1 by 4, greater than 1 by 4 we observe two recombinant techniques. Okay, this is test crossing progeny. Say which one less than and which one is greater than we observe. Okay, types of linkage genes on same chromosome and tends to gather during the formation of gametes, does not assert independently. Unlinkaged genes on different homologous chromosomes assert independently. Okay, next coupling and repulsion hypothesis. Coupling and repulsion hypothesis, we observe this concept as given by Bateson and Punnett to explain the law of independent assertment in the result of either experiment, but they could not explain except reason. Okay, later re-explained by T.H. Morgan. Okay, first one, coupling phase, when two linkage genes on each chromosomes are the same type, is also known as cis phase. Parental type, that is both dominant capital A, capital B, or some both dominants are absent, or capital A, capital B, or both recessive or small a, small b. Repulsion phase, when linkage gene on each chromosome were different type, is also known as transphase. This cis phase is both dominant capital A, capital B, and both recessive small a, small b. We observe this cis phase. This recombinant type is different. We observe this capital A, small b, or some this option, but small a, small b mistakenly formed. Okay, recombination of linkage genes occur in same frequency. Okay, capital A, capital small b, capital A, or small b, recombination of linkage gene occur in same frequency is transphase, and is called recombinant type, and same is formation recessive, and domination is a cis phase. We observe this. Gene mapping. What is gene mapping? Linkage of genes in chromosome can be represented in form of genetic map or linkage or chromosomal map. First chromosomal map as successfully done of Alfred Sturtevant. Okay, Alfred Sturtevant in Drosophila using gene recombination frequency. Recombination frequency, how to identify gene mapping is called total number of recombinants into 100 by total member of progenesis. Okay, we have progenesis is gene mapping, total number of recombinant into 100 by total member of progenesis. Gene mapping is Alfred Sturtevant in Drosophila using gene recombinant frequency. Okay, this we observe 49 meiosis with no crossing over. 49 meiosis. Okay, this type of chromosome. So um, um, we will be come to same color, okay, blue blue, red red, okay, it are not crossing over, no crossing over. So 49 non recombinant we observe 4, okay. Next B diagram we observe frequency of recombinant in second diagram, okay, one meiosis with a single crossing, okay, we observe one meiosis with a single crossing over. How it form? One total is non-recombinated, second one recombinated, and recombinated we observe, and the last one also one non-recombinant. Next, frequency of recombinated, how it gene mapping is present. First, R is equal to means recombinated 1 plus 1. We observe 1 plus 1 is the recombinated. So we observe this yellow color is marked 1 recombinated and second one recombinated. So recombinated member we observe 1 plus 1 by 49. We observe this non recombinated 49 49 49 49, both are plus to add, then it plus plus 1 plus 1 plus we added 2 by 200 into 100. We observe one parent, one map unit is equal to 1 centimeter. So this type to use to gene mapping. Next, features of genetic recombination. Gene mapping determine the order of genes and the relative distance between them in gene units. One map unit is equal to 1 cm means centimorgan is important to centimorgan. Gene mapping method use recombination frequencies between alleles in order to determine the relative distance between them. Recombination frequency between genes are inversely proportional to another distance apart. Distance measurement, one map unit is equal to 1%. One map unit is equal to 1% recombination. Okay, true for short distance. Okay, genes with recombination frequency less than 500% are on the same chromosome is called link recombinates two genes that undergo independent assortment having recombination frequency of 500% and are located on non-homologous chromosome or for apart on the same chromosome, unlinkage parental type. Gene mapping crossing over. Okay, exchange of genes between two homologous chromosomes is known as crossing over. crossing over which occur outside the region between two genes will not alter their arrangement. The result of double crosses between two genes is indistinguishable from independent assertment of the gene. Crossing over involving three pairs of alleles specify gene under linear sequence of genes. Okay, this to observe two strands double stranded. Okay, this was crossing over gene is 4. Okay, 4 genes are recombinant gene we observe. This is also double stranded crossing over, and the 3 strand crossing over, double stranded crossing over, and 4 stranded crossing double over. So we observe this. Okay, this is parental recombination. Okay, parent 4 recombinant is 0, and parental 2 recombination also 2, and this we have some parental and recombination 2 we observe. This parental is 0 is a 4 stranded double crossing over. This 3 strand double crossing over and 3 strand double crossing over we observe. Okay, recombination is 4 stranded, so we crossing over 4. This is total. Parent is 8 and recombination is 8, so it come to 1:1. Okay, another point we go. Okay, this E.H. Morgan linkage in Drosophila. The first genetic map construction in the organism, such as the fruity fly genes and markers. Okay, we observe this. Okay, gene mapping, linkage mapping, and physical mapping is closely and distinctly related, and chromosome and this phi and X. Okay, we observe. Okay, next crossing over. Okay, crossing over also discovered by T.H. Morgan. Recombination of a gene on the same chromosome. Parts of homologous chromosomes interchanges. Okay, this homologous we observe the homologous chromosome interchanges. So this crossing over for product of meiosis we observe this. Already we know it. Okay, the form to non-recombinant chromosome, recombinant and non-recombinant chromosome. So it crossing over is called a recombinant chromosome. This also same. Crossing over occur in Mitotic division causes loss of heterozygosity. Okay, heterozygosity. Loss of heterozygosity uh, is L means loss, O means loss of heterozygosist is a gross chromosome event that result in loss of the entire gene and the surrounding chromosomal region. This crossing over is essential for normal segregation of chromosome during meiosis. Crossing over also occurred for genetic variation because due to the swapping to exchange something for another of genetic material during crossing over. Okay, this uh, percentage of crossing over, how it form is crossing over percentage, the percentage of total member of aspirin in F2 and total member of recombinant into 100. Okay. Okay, some multiple choice questions also there. Okay, percentage of crossing over in more than linkage gene are located for apart from each other. Okay, linkage gene are located for upper from each other. Okay, if the percentage of crossing over between two genes is 10, then the distance between two genes will be 10 centimorgans. Okay, very important. 10 centimorgan is very important questions. If the percentage of crossing over between two genes is 10, then the distance between two genes will be 10 centimorgans. Alleles of different genes that are on the same chromosome may associatiously will be separated by a phenomenon known as crossing over. Okay, yes. Crossing over in diploid organisms is responsible for recombination of linkage alleles. Okay, thank you. Okay, next go to another slide. Okay, we observe crossing over genetic recombination. Okay. What is crossing over? The exchange of chromosomal segment between two non-sister is very important. Two non-sister chromatids. Okay, crossing over is important. When does it happen? Okay, meiosis increases genetic diversity in a species. Mitosis create genetically identical daughter cells. Where does crossing over occurs? Genetic swapping occurs between paired homologous chromosome in over sex cells, the egg and sperms. Egg and sperms. Chromosome pair. Chromosomes may swap genetic material. New genetic material has been acquired from the over homology. Note: some genes that have a tendency to remain together during crossing over are said to be a linkage. Uh, we will talk about this except during crossing over basics. Okay, this exchange DNA by crossing over to provide genetic variations during meiosis. Okay, what's why do and I do not look alike? Why? Crossing over ensures a combination of a maternal and parental genes we inherited. Okay, the history of crossing over. Okay, we observe Gregor Mendel 1823 and 1884, patterns of inheritance. Carl Corans in 1900, gene linkage is Alfred Sturtevant is important. The Gene Mendel, Gregor John Mendel. Okay, next, crossing over basics. Crossing over basics occurs at one or more points along are just homologous during synapsis points except each other DNA is exchanged. Okay, we observe this is called DNA exchange is crossing over to form the gene mutation. Okay, recombination, crossing over and recombination during meiosis. Okay, this white and black we observe dot, then it formed to crossing over in the gametes also form. Okay, crossing over basics, gene mapping. Okay, this we observe. Okay, task in crossing over help to determine where genes are located on the chromosome. Genes that are far apart having greater change of crossing over. Genes that are closer have the less linky change of crossing over. Genes that stay together are uh, said to be linkage. One gene can be identified as a marker that can infer the presence of the another gene. These can be used in identifying disease, crossing over basics, gene mapping. Okay, tracking crossing over help determine where genes are located on the chromosome. Genes that are for a part have a greater change of crossing over. Genes that are closer have a less link change of crossing over. Gene that okay, this animation to show that how it shuffle when chromosome exchange pieces. Okay, okay, this was a summary how, what, when, where, why, history based example and summary, we all know it. Okay, okay, thank you for giving this time. Bye.